Okay, I think that we'll just call this a quorum, and I'll go through some introductory remarks that people could probably skip anyway, and then we'll have Dan start. So hello, everyone, and welcome to the inaugural meeting of the Scientific Visualization Interest Group. This interest group will explore the theory and the practice of making pictures from scientific data. So although we intend to discuss some widely used tools, we thought we'd start off with a series of talks on lesser known and newer technologies that may be very useful to our scientific mission at Genelia. So in particular, how do we extract the most information out of the extraordinary imaging data sets that are being produced? Computer graphic software and the hardware behind it is really progressing very rapidly. And so we want to make sure that the visualization software is as state of the art as the imaging that we're now getting. And so with that in mind, I reached out to Dan Toloudis after reading about his work on the Agave system. It's a nice example of advanced rendering techniques used for scientific imagery. So Dan is a senior software engineer at the Allen Institute. He got his bachelor's degree from Johns Hopkins and his master's from University of Oregon, both in physics. And he currently specializes in graphics. And rather than me talk about his background, I'll just turn it over to Dan, and he can show us some of the great stuff that he's doing.
Okay. I will switch to screen share. So thanks, Bill, and thanks for inviting me to speak today. My name is Dan Toloudis at the Allen Institute for Cell Science. I'm a software engineer working on the animated cell team. And today I'm going to talk about volume visualization and rendering in the context of biological data and the tools that we developed at the Allen Institute for Cell Science. So to give some context around what I do, the overall mission statement of the Allen Institute has two parts. And the first part is to understand how cells organize themselves and change. Toward this end, we capture 3D microscope images of living cells. The cell lines we produce have been gene edited to produce fluorescent proteins to prepare them for imaging. And we capture large batches of images under a variety of different conditions. So the animated cell team that I work on is closely tied with this second part of the mission statement. We want to develop and democratize tools. So what that means is making the Institute's results accessible, usable, and interesting to the outside community. So most of what I'm going to discuss today is in the context of tools I have worked on for making our data more accessible. So as for myself specifically, I've been developing tools for interactive 3D graphics for basically my whole software engineering career. So little disclaimer, I'm not a biologist. My background is in physics, but I am basically a graphics software engineer. So I worked on everything from office furniture layout tools to product marketing visualizations to animation tools for entertainment industry. And all of the images here were rendered with code that I wrote. So now I'm working on 3D visualization tools for scientific data. So to just quickly orient ourselves to the type of data that we're dealing with now, microscopes capture intensity values in a two-dimensional focal plane. The data is stored in sequential slices. So the focal plane is moved up and down the sample being imaged. Each slice is considered to span an XY plane, and the collection of slices runs along the Z-axis. So we often refer to it as a Z-stack. Just so that because the Z-axis is going to be prevalent in a lot of the imaging modalities or the visualization modalities I'm talking about. So when I say Z a lot, you're going to know that's what I mean by Z. And the other thing that's interesting to note is that the values captured by the microscope are typically just intensities. These are just single numbers, scalar values. So we're basically dealing with a 3D brick of numbers, and we have to turn that into a picture. So we want to examine these images now with the naked eye on a computer screen. So this is a qualitative examination. We can count things, but we are fundamentally making qualitative assessments. We're eyeballing these images, looking to see what shapes we see. This is what biologists have been doing for centuries. So I'm going to talk about these 4 different ways of looking at volume data, sort of at a high level. As an example, I'm going to use this particular cell from our integrated mitotic stem cell data set. And these 4 approaches I'm going to talk about, they all exist in many different volume viewers, but I'm going to describe them in the context of the tools that I have developed for Allen Institute data. One thing to note about volume data that's common to all of these approaches is that the intensity values captured are generally spanning a large dynamic range, and they can also be noisy. There can be non-zero intensities in areas that are not the thing we are actually trying to image or that we care to look at. So we often have to remap these intensities into ranges that make sense to be visualized, to be displayed on a computer screen, and that will make sense to our eye. And that includes doing things like changing contrast and removing noise. There's basically always some type of intensity adjustment that has to occur for display. And that particular adjustment may be different for each of these viewing modes. So yeah, these links actually will take you to that particular cell on our website. Okay, so the simplest approach, Z-stack viewing. This is a 2D approach. This is simply, we're going to look at one slice from our volume data at a time. So this is great when you want to look at absolute pixel intensities because you know that you're just seeing the exact pixels that are on that slice. This is great for isolating layers where something interesting is happening. It does require you to assemble the 3rd dimension in your head. So the typical use case is, you will look at one slice, then you will look at the next slice in Z, and then the next slice and the next slice. And often we play them back in a movie as a time sequence, but really, and so it looks like a movie, but what we're really seeing is a cross-section moving up and down in the Z direction. So a classic problem with this is that you can imagine like a thin fibrous structure that starts in the top corner of your Z stack and goes diagonally down to the opposite bottom corner of your Z stack. And if you were viewing this slice by slice, you would just see a dot that appears to move across the screen as you iterate through the slices. And so your brain has to interpret that. You kind of have to fill in the blanks there to actually perceive a fibrous structure that's doing that. However, this is like commonly used. It's sort of like tried and true method. Everybody does this. This is the simplest, most basic way of examining our data. So here's just an example from our integrated mitotic stem cell. This is a Z-stack viewer where we're looking at a channel of interest. And then in the right-hand panel, we have that channel of interest in context with the DNA of the cell in cyan and the cell membrane channel in magenta. And here's our Z slider. So we can we're selecting the actual central slice of this volume. And of course, we can do things like move the slider up and see a different slice, move the slider down and see a different slice. So this is pretty ubiquitous in volume viewing. A second way of examining volume data is to use maximum intensity projection. So this method involves calculating the maximum intensity for all Z for each XY position. So the result of this is a single 2D image where at each pixel we've selected the maximum intensity among all of the Z slices at that position. So this is a pretty common way of representing the most important features of a Z stack in a single 2D image. If you wanted to present your volume data as a single image, you would often choose either a single slice that's representative of the important data or a max projection. So this works really well for high contrast images where the important thing you're imaging actually lives in the higher intensities and it's separated from background or sort of non-signal in lower intensities. It is important to note that the third dimension is completely gone. We are removing a dimension by virtue of selecting only the maximum value. And this works great when there is less superposition of structures along the axis of projection. So if you imagine two objects being imaged and they're like on top of each other in our Z stack, if one of them were much higher intensity than the other, then the lower intensity object would just be gone from our max projection because only maximum values were selected. And if the two objects were similar in intensity, then they would appear to be just merged together in this image, and you would have no information about which one was on top or on bottom. So again, it's a 2D approach. So here is a max projection mode in our Cell Feature Explorer web viewer. So the Cell Feature Explorer is a tool that we have that's online that presents a large data set of like 200,000 plus single cell images. And it's a 3D viewer, but it has a maximum projection mode. So the interesting thing is that in this viewer, you can rotate the volume in 3D and still do maximum projection along the viewing axis direction. So you can orient the volume in a different direction and still get a maximum projection. Again, it's important to note that this is deceptive for spatial relationships along that axis. So slice and max projection, again, are 2D modes of viewing. The first true 3D mode I'm going to talk about is an algorithm called ray marching. So this is sort of one of the simplest but reasonably good quality approaches for getting 3D volume viewing in an interactive viewer. So the algorithm works this way. At each pixel of our viewer window, we're going to march through the volume, sampling the intensity at discrete steps inside the volume. And the resulting output depends on how we combine these samples. So like I described before, we can do a max intensity projection in this way just by selecting the maximum value of all the samples along that ray and discarding the others. But we can also start writing more complicated ways of accumulating the intensity or representing transparency. One simple, obvious thing is we can just stop marching the ray when we reach a sample that has a high enough intensity, which means we would basically consider that region of the volume opaque to anything behind it. And so we can terminate the ray marching and just return that value. In practice, we do something like that where we have a termination condition, and we also do something like an exponential scattering absorption function, where it's exponential decaying with distance traveled, also proportional to the intensity at the scattering points. This is Beer's law from physics. And now we're starting to introduce elements of physical light behavior and starting to treat the volume like a light scattering medium in order to start getting more realistic views of it. So using that Beer's law decay, we can build images that are that have sort of proper transparency and proper depth. So here's our Cell Feature Explorer's ray marching volume viewer. You can get a more immediate sense now of things being in front of other things, and you can sort of trust that when you know you're in a volumetric ray march mode. Things that look like they're in front are in front. And this is much more apparent when we rotate the image interactively. So here's a little screen recording of what that interactive rotation looks like. So you really get a sense of the three-dimensionality of the structure by tumbling and looking at it from different angles. This kind of interaction almost encourages a dialogue with your image where you're examining it while tweaking settings to sort of highlight or pull out interesting features that you notice as you're manipulating it. So the next jump in complexity comes when we want to add lighting, shadows, and surface reflections. So the purpose of this is to make spatial relationships in our 3D data even more apparent at a glance. So I'm going to describe the path tracing algorithm at a high level over the next couple of slides. So let's start with a simplified ray tracer. If we have a scene of objects made of hard surfaces, like this sphere, we can begin to add natural looking lighting in this way. We send rays into the scene, and when they hit an object, we calculate a bounce direction toward a light source. And we see whether that bounced ray hits anything before it reaches the light. So if a bounced ray makes it to a light, such as in this uppermost intersection point, then we know that ray is going to contribute color to our image. In other words, some light was reflected toward our camera. If the bounced ray hits another object before it hits a light, as in the case of this point where we trace our ray toward the light and the sphere is in the way, then we know that that point is receiving shadow and that light source is not contributing any illumination on that point. So just by doing that simplest version of ray tracing, we can build a scene, we can build images that have shadows and lighting into them. Now, to make this more realistic and more physically plausible, we can introduce a more rigorous way of calculating the light arriving in our camera. So this equation is known as the rendering equation, and it forms the basis of almost all 3D computer rendering done today. Almost every 3D display technology can be treated as some approximation of this equation. This equation is very closely related to the radiative transfer equation from physics. So we're actually modeling light transport at this level. So I'm going to just give you a high-level interpretation of the terms in the equation for understanding, but we're not going to go any deeper into the math than that at this point. So the equation is interpreted like this. The light directed outward from a point X in a given direction is equal to the emitted light from that point X in the same given direction, plus the sum of all light incident at that point that scatters in the given direction. So this sum, so the F term in this integral is a reflection distribution function that describes the light scattering response at our point X. So it's giving us a proportion of light that scatters in a given direction from a given incident direction. So this basically describes the sort of surface appearance of our surfaces. This is our, in a sense, it's our material description. And the outgoing light term LO and the incident light term LI are actually the same quantity. And so this is, in that sense, this is a recursive integral equation. For practical purposes, that means it's very hard to solve. And when we do this in practice, we have to use stochastic Monte Carlo methods. We're going to basically randomly sample the L function and converge to the solution by increasing the number of samples. So the number of ways to try to make this efficient and tractable is huge and the subject of vast numbers of research papers and whole conferences. So for our purposes, we're going to think of it this way. We can trace a single ray, and in order to figure out how much light is coming from the first surface point to our eye, we really need to integrate over all incoming light from all directions that impinge upon that point. And if we just think about one particular direction, if that ray reaches another surface, we have the same problem. The light coming from that surface is really the sum total of all incident light reflecting off of that point. And so we can basically treat this red path as this sequence of red bounces as a path, a single path. So we only get light contributions from paths that go between camera pixels and light sources. If a path doesn't terminate in a light source and just exits our scene without hitting anything, then it's going to have a 0 contribution to our final result. So in this way, we can actually do this one path at a time by randomly choosing directions. And eventually, this is randomly sampling our L function. And if we build up enough of these paths, we're going to converge to the correct result. So for our volume data in practice, this takes a few seconds to converge, but the actual time to converge depends on a myriad of factors. For example, it depends on the size of the volume, the number of pixels in our window, the number of ray bounces along the path, the complexity of the reflectance function, the size and intensity of the light sources, which is related to the probability of a random bounce actually hitting one of those light sources. Many other factors, obviously compute power available also. But the important thing is that if we can actually trace one path per pixel at interactive frame rates. And so what we can do is we can actually build up the image interactively. And like I said, it takes just a couple of seconds before the image comes into focus. And I'll show a live demo of that so you'll see exactly what I mean. So that was a general explanation of path tracing. And here is the specific version of path tracing that we use for volume viewing in our tools. So the strategy is that we're gonna simplify this quite a bit. We're going to trace a ray, for example, this blue ray, and it's going to reach our volume. It's going to start, we're going to march it through the volume according to the ray marching algorithm. And we're going to sample intensities along the way. And we're going to do a stochastic evaluation as to whether a scattering event has occurred or not. So again, this chance of scattering is proportional to the intensity values in the volume data. Higher intensities are more likely to scatter light. Lower intensities are more likely to allow light to pass through. So if we determine a scattering event has occurred, we're going to cast out 2 rays, one ray toward a light source and one ray whose direction is chosen based on our reflection distribution function. So we're going to sample that reflection distribution function and just allow the ray to reflect in a way that is proportional to that reflection function. We marched both of these rays through the volume in the same way. We marched and sample intensities. And if we determine that a scattering event would occur along these rays, I just terminate the ray. And I consider that ray to have 0 lighting contribution. If the ray actually makes it out without scattering, then we can collect light from any light source that ray reaches. So what that means is the approximation that this approach is using is a single scattering approach. In reality, there will be multiple scattering events within the volume, and rays would zigzag and bounce around many, many times before they even exit the volume. So this is another sort of approximation that makes things tractable. And it's kind of like, you can think of it as keeping the first order effect and discarding higher order effects. So with these approximations, we can actually implement this in a web browser. Here's what it looks like in the same cell feature explorer viewer with our same cell. So we're immediately getting lighting and shadows just by the nature of the algorithm. One thing that makes this tractable for web browsers is that we have downsampled our volume data for display in browsers. This is actually a huge optimization and lets us really run this type of code in any web browser and get reasonable performance. We've even tested this on cell phones to make sure that it can run interactively. And to simplify the user experience for this kind of presentation, we fixed several of the parameters. In particular, the light source positions and the intensities of the lights are just fixed for this display. So in our desktop viewer, Agave, the volume data is not downsampled. So what you're seeing here is from the full resolution original raw microscope data. Because of this, Agave has higher compute requirements, and basically, the better the GPU you have, the better it will run in terms of both speed of GPU and amount of memory on your GPU, because we load the whole volume data into GPU memory. And of course, in this tool, users have full control of the light intensities and positions, and they can control some of the reflective properties of the volume also. So in this particular case, you can see this magenta colored channel has these brighter, shinier reflection highlights compared to the other two colored data channels. So the thing to note here is that you can get a very good sense of spatial relationships from the well-defined shadow and lighting cues. Even the reflective highlights are kind of indicative of the shapes that we're seeing, and there's less of a need to tumble or rotate this view to know what's going on. But you can still do that, of course, in the viewer. So here's just another screenshot of Agave running on Windows. I think the previous one was taken on a Mac. And I'm showing a larger volume data in this one. This is actually the original microscope image from which that same cell came. So that same example cell is this one right in the middle of our view. Just an example image here. This is characteristic of the sort of size and scope of the image fields that we capture. Here's another capture from Agave that shows that you can apply interesting color choices. And because of the path tracing algorithm, and because it's doing ray optics, there's also a physical lens model built into the algorithm. So we can control parameters like lens aperture and focal distance. And we can create these depth of field effects where far regions and near regions are slightly blurred. They can be blurred considerably more than this, of course, but you can choose to defocus these areas if you want to, for example, force the viewer's eye to gravitate toward a certain region, or just to make it look fancy. And of course, we can also display derivative data from our raw data. So this is the result of a segmentation algorithm. Essentially, this is just a binary volume data set where every volume pixel is either filled with a 1 or a 0. And if we embed these channels in the original image, we can easily just flip them on and off in the interface, or you can do things like switching. You can generate images like this and quickly switch back and forth between them to sort of compare your segmentation results with your raw data. We can also script Agave to produce animated sequences. So this is an example with camera rotation and also with animating a color from red to green. This was basically built with a Python script that just does simple linear interpolation of values and re-renders an image. We've also done this with pretty long time lapses as well, and I'll show one of those later on. So lastly, or nearly lastly, I want to just mention some of the challenges that make this all difficult. So first of all, to display multi-channel data requires pretty careful selection of how the channels are combined together. This is surprisingly non-trivial, even in the 2D case. The order in which you combine things can matter, and the weights that you give each channel make a difference. And in the 3D case, we have choices of different strategies. For speed reasons, we chose to take the maximum value among all the channels at a particular volume location when we're sampling. But an equally reasonable approach would be to just randomly choose a channel to choose an intensity at the sampling location. I haven't tested this yet. I'm certain that it would cause images to converge more slowly, but the question in mind is whether it would give more accurate overlap of structures that are coincident with each other in the volume. So another challenge is dealing with very long time series data. One part of that challenge is simply load times of very large files, but we can kind of handle that by knowing where in the file is the particular time sample that we care to load. And that kind of basically solves that problem. However, the larger problem is when there are intensity fluctuations across the time sequence, and we want to just write a script that can go through the whole time sequence and render out an image for each time. In order to actually try to normalize intensities across the whole time sequence, you effectively have to load and walk through all of the data in advance of even rendering it. So as I mentioned before, for the web-based viewer, we downsample our data. So, it's possible to produce very large volume data sets, even for a single time sample. And downsampling the data really allows fast interactive viewing. It is a good way to guarantee that it's going to fit in memory. But for Agave, we really want to render the data at full resolution. And this means currently that we are limited by GPU memory without doing any more clever tricks yet. So there are tricks. If you have sparse volume data, which means that there are regions, significantly large enough regions where the values are constant, then you can apply various compression schemes to it. And some of these compression data structures actually not only optimize the memory that it consumes, but they can also be used to optimize the ray marching across those constant regions. And of course, there are also ways to deal with large volumes by splitting them up and loading the subvolumes on demand. These tend to be slower and not optimal, but there are ways to actually allow this type of visualization when maybe it couldn't happen otherwise. So another challenge we face is that many of our users want to automate the rendering process. We generate lots and lots of microscope data. We have automated processes that just capture image after image. And so, for example, our scientists may want to generate several renders of a list of different cells with the same rendering settings. And the problem is that really the best results require some hand tuning of these settings right now. Figuring out how to do automatic thresholding is pretty hard for this type of 3D path trace rendering. We suspect that there is probably an ML approach that can improve this. Machine learning denoising is pretty well accepted now for 2D imagery. So the question is, can it be applied to volume data? So the way our users would do this today is you would actually do an interactive session in the interactive viewer. You would dial in all the settings you like and basically save them out to a Python script. So the Python script will be pre-populated with all of the settings that you've chosen, and then you can modify that script to run through a long series of files and apply the same settings. In practice, these settings are actually pretty sensitive to small intensity or noise changes in the data, so you can get mixed results from this, but it is a solution that we would use. So another challenge is just certain types of images. You also can get mixed results when you have noisy data with small punctate structures or wide diffuse fuzzy structures. Lighting effects can be lost with these types of data or have less impact. So, the bottom line is that high signal to noise ratios and well discernible shapes will always lead to easier and better volume rendering results. And then lastly, so my team, the animated cell team at the Allen Institute for Cell Science, is interested in multi-scale viewing of cells and processes within the cell. And we are interested in combining volumetric data with surface meshes, fibers, sphere-based molecular representations. And we have also built a web viewer to display time series agent-based simulation results. That's called the Simularium project. And as part of that project, we are hoping in the near future to introduce volumetric data into that world so that we can combine the two. So just a couple more images of representing some of these challenges. Here's an image that was captured with really good signal to noise. And it was really easy to threshold this data. In fact, if I remember correctly, I barely had to touch any settings. The data just came in looking like this. So I just moved light sources around to cast light and shadows to make some of these bumps apparent. But really, this data just, if you have nice, clean data coming in, you're going to get very good rendering results. This is an example of what the current Simularium viewer looks like. This is some kind of endocytosis, invagination of the cell membrane. This is just like one example of many, many different types of agent-based simulations that we can represent in this viewer. And so you can see how seeding this type of simulation with real volume data could potentially like enhance not only the simulation, but also the visualization of it. And then here's an example of a longer time series that we captured and rendered with Agave. There we go. So it's kind of subtle here, but the shading and shadows that you see where the nuclei come together are really bringing out the three-dimensionality of this. And they really make it sort of, it just looks less flat and more realistic. And we're getting a true sense of what the shapes of these things really are. And this is just an example of a very long time series. So all of the tools I've described are open source, and they're all available through the allencell.org website. And I want to also say that I'm available by email to discuss any of these things. And you can download and examine the code. We welcome contributions, issue requests, users. We'd love to see what anyone is doing with this stuff. And with that, we'd like to thank our founder for his vision, encouragement, and support. And I'd also like to thank the director of the Animated Cell team, Graham Johnson, for his continued support of this 3D visualization work. So with that, we can do questions. I can also do an interactive demo of Agave to show you how it looks like in an actual user session. Up to you guys. What would you?
Ricky has a question. Did you want to hold that until after the demo, or do you want? Oh, you're muted. Sorry.
I can wait until after the demo. Then we have enough time that we can enjoy the demo. That I can ask the question later. That's fine.
Okay, Dan, you can go ahead with the demo, and then we'll do the Q&A.
Sure. Okay. So what I'll do is I'll just launch Agave, and I'll just load a volume in and start playing with the sliders, basically, show some of the effects you can get in the program. Let's see. One moment. I will screen share. Okay, so hopefully you are now seeing the Agave interface with nothing loaded yet.
Yep.
And I'm going to open a recently loaded one. So I'm going to load a larger volume, a full field of cells here. So this is an image that you can download from the Cell Feature Explorer website. Oh, you know what? I don't know how this is going to look on your screens, but I'm going to stretch my user interface to widen it a little bit. So these images that we provide have 9 built-in channels. 4 of the channels are raw data from the microscope, and the other 5 are derived data, segmentation data, essentially. So you can see when the image comes in right off the bat, we try to apply some thresholding settings to it. And you'll also notice, if you're getting good enough frame rate over the internet, that as I rotate, the image becomes very, very noisy, right? That's because of the path tracing algorithm only being able to trace one path while I'm moving the camera around. So some of those rays are just not picking up light and some are. But then as soon as I stop, it keeps iteratively tracing more and more paths. So the image resolves fairly quickly. So the initial settings are not perfect, of course, for this data. So there are a couple of big sliders that kind of affect the global image appearance. The first one that is interesting is scattering density. So if I move that up, basically, this is just sort of like, it's roughly increasing the intensity of every pixel in the image. The effect of that really is making everything look like a hard surface. And if we decrease scattering density, then everything becomes much more translucent and sort of wispy and cloud-like. So what I'm going to do is for this, I'm going to kind of turn that up pretty high, and then I'm going to dial down. I'm going to threshold out some of this DNA data. Percentile. Yeah. Basically, what I want to do is I know that that is the nucleus channel, so I want the blue areas to look like nuclei. And what I should do while I'm tweaking that is turn off this white channel completely. So this is just now looking at the membrane and nucleus channels. So these are looking pretty round. This one is still really fuzzy, and that I know that's because this is a much higher intensity because that's our dividing cell. So if I threshold that data some more, we'll bring that into really good focus. So we're getting a sense of like just how much more intense that area is than the other areas. Just turn that back a little bit. So I'm going to bring back the microtubules channel and threshold that a little bit. To bring out some of those fibrous structures. And then I'm going to show one of the some of the lighting manipulations we can do. So what I'm going to do is I'm going to make them a little bit shiny. So this is just going to increase the reflectivity. So I'm going to change this specular color and this glossiness value. I'm just going to increase the glossiness. I'll just put it at maximum for our purposes. You don't see much of a change here yet, and that's partly because the light source that is dominating right now is a spherical light that's surrounding the entire image. So light is impinging from all directions, and so it's hard to find a preferential reflection direction to actually see bright reflection highlights. But if I sort of amplify that effect by going to the lighting, so we have two lights basically. You have only two lights to play with here. The skylight is that sphere that's surrounding the scene, and the area light is a single light source that you can position anywhere in the scene. So to see the lighting effect really more dramatically, I'm just going to turn off the skylight by setting its intensities to 0. Now immediately I'm seeing those directed reflections from my area light source. And if I just move that light source, you can see the shadow direction changes and the reflection highlights change their location. And so I'm basically just controlling angles that are moving that light source on a sort of, if you imagine it as a square-shaped light that's moving around a sphere above our scene, above our volume. And we can control how far away or close it is. If we bring it close up, of course, everything becomes very, very bright. If we move it farther away, things get dimmer, and we can control its intensity. So if I were producing an image, like for an illustration, I might use more dramatic lighting. But just for like ordinary sort of examination, the skylight is generally like pretty sufficient, and it very quickly dominates. And of course, the other interesting thing is you can change colors of all of these. And the skylight has 3 color choices, sort of the North Pole color, the equator color, and the South Pole color. So I'll just dial in some arbitrary choices so you can see the kind of effect it has. So it just gives the image an immediate change in quality. Another interesting thing to note is that it's continually re-rendering and adding path samples while we're sitting here. And a lot of the time I will be in an interactive session and I'll maybe get up from my desk and walk away and come back in a minute. And there will be a subtle but really interesting quality change in the image. So, samples that happen later contribute less to the image just by virtue of the way they're averaged in. So it takes a little bit more time to see changes. So it can be really interesting when you walk away and come back, and then suddenly the image just looks just a tiny bit sharper or better or more converged. I can also demonstrate what the field of view settings look like. So there's no performance impact for those. It's just part of the path tracing algorithm. So if I change my aperture away from 0, we immediately will have defocusing effects. And that's all you have to do to immediately see it. And then you can change that region of focus just by changing the focal distance. Oops, not to that number. I'm going to bring everything back into focus for now. Let's see. So you can also do orthographic viewing. If you don't want perspective foreshortening like this, you just switch to an orthographic mode. This is just preserving parallel lines. There's a bit of an optical illusion now where it looks like these lines are converging, but they're really not. All these lines on the same axes should be parallel in this mode. Let's see. Yeah, there are a few other just viewing parameters. You can turn bounding box on and off. You can actually change the background color. You want to put your image on a white background. And there are buttons to save the image out. If you want to preserve your settings and the current image that you're working on, you can save to a JSON file. And then that JSON can be loaded back in later. You have to be careful if you do that because it's going to save the absolute path to where your volume data lives. So if you ever move that, you have to either hand edit the file or else it's going to be useless. And of course, you can save to a Python script. That's a pretty good overview. There are a lot of more fiddly settings. I think that's kind of a good summary of the main features and sort of how I would start to use this tool. It also helps a lot when you have some of this interesting derivative data. Like I can turn on this contour DNA channel and turn off my existing DNA channel. That's this one. And let me change the color. So now if I zoom in, I'm kind of mixing the this calculated data with my raw data at the same time. So those are contours around our 5 segmented nuclei in this data set. Is there anything you're curious to see or that you'd like me to demonstrate in addition? I'm happy to take questions or just have open discussion at this point.
Yeah, so I think Ulrike, you want to ask a question now? Yeah.
So I was just wondering, so the tool is called like Volume Explorer because you want to use this in order to actually inspect your data as well. But let's say right now you set things up in such a way that you can actually see your data in specific areas in a better way, and then you actually recognize, oh yeah, that's actually something really which I would like to even quantify further. So my question actually would be, is there an option in that tool also to specifically maybe even crop out a region and export this as well? Because I think right now it's focused mainly on the visualization part and less on the quantification, meaning in order to quantify a certain part, you actually would need to export that again. And then kind of is this also part of the tool as well?
So
it's like cropping and exporting of a cropped set where there might be.
Cropping for visualization is possible. That's built in here. So we can just move these sliders and start to construct regions. Export is not an option here. The tool is really sort of an endpoint for visualization, and the output of the tool is currently intended to be just pictures. It's a big discussion we've had. We know that a lot of these tools also build in analysis features and quantitative analysis features, and it's sort of a matter of where we want to evolve the tool, and user feedback helps drive that too.
Yeah, because I do.
Sorry, if you modify the ROI, does that go out in the saving to the JSON?
Yes.
So that you can, okay. So you can record the ROI then that way.
Yes. Yeah. So if you wanted to share this JSON file with a colleague who could then run Agave on their own workstation, for example.
Did you have any other aspects of the question, Ulrike, or?
No, that was in principle. I mean, because the thing is, like, as I've mentioned, one can have this as a final publication image, but it would also be great to have it like as you mentioned, like as an inspection to maybe then continue to do the quantification again. I mean, I think that's sometimes quite useful. But anyway, yeah, that's it.
Thank you, Stefan. I think you were next.
Yeah, I mean, it was a pretty cool tool, to be honest. I was wondering, since you can also make like really nice animations, is there any way to distribute that? Say I want to make a nice video of 1,000 time points or a very large volume, and I find my parameters, and I want to do that. Is that possible right now?
So currently, the way that you would do that is very primitive, and you kind of have to do it yourself. So the way that you would produce an animation sequence right now in the tool would be via Python, and you would essentially output one RGB image file for every time in your sequence, and then you have to use your own devices to cobble that together into an MP4 movie and distribute it you're kind of on your own to do that distribution.
I mean, that's easy to do. And how about bigger volumes? Because then it's not so easy anymore, right? To tile things. Let's say I have like a 10,000 by 10,000 pixel or even bigger image, and I want to make tiles there. Is there any way to do that too?
There currently isn't without pre-processing the volume data. So you are limited by GPU memory right now, and the in-memory representation of the volume data is 16 bits per pixel or per scalar intensity value. So there's a little log file that Agave produces that I'm not showing in my screen share, but it kind of gives you some information about how big the volume is that you're loading. And it will tell you if you try to load a volume that's too large to fit in memory. But currently, right now, the tool is a little bit, you could say, in a primitive state where it's only going to work if the data you're trying to load fits on the GPU.
And maybe I said it and I missed it. What are your big future directions with this tool? You want to maintain it at the current state, or you have plans to go in a certain direction from here on?
I think one of the big ideas right now is to integrate this with the molecular simulation viewer. We want to kind of bring together other types of geometry into a tool like this and be able to generate really high quality renderings using the same path tracing approach, but with other types of geometries. I mean, another common thing that people will do with volume data is generate surfaces from objects in the volume data, and being able to render those things as triangle meshes is sort of a natural. Yeah, I think I would say that for Agave itself, it's going to undergo an architecture update, which probably won't change any of the functionality, but will make it run on a wider variety of machines. For example, if you want to run this like on a headless cluster, there are some limitations with the way the graphics library is initialized, where on some platforms, it really requires you to have a window on a display device in order to initialize it at all. So there's some sort of technical problems like that under the hood. But really, I think that user feedback will drive the future directions of what we add to this tool. So like if everybody says the image quality is great, don't touch it, but we need to be able to export sub-regions of the volume. I just heard that today. You know, if we hear that loudly enough, then that becomes a feature that we will implement.
Thanks again. Very nice talk.
Yeah.
Thanks, Dan. Philip?
Beautiful images, Dan. A question I have would be, how would you compare it to some other systems that people might use for generating images? Just as an example, people at Janelia like to use Blender, and I understand. I could see that one comparison would be that Blender doesn't have the highly tuned user interface for the interactive viewing and setting the specific parameters of interest for this particular kind of rendering. What are some other ways you might compare the advantages of your system to some other systems like that?
That's a great question. So I think that the one that you named really is the major differentiator here. It's that we want to read in biological data as easily as possible. You just hit file open and it loads the data. And all of the user interface controls are tuning specifically the appearance of volumetric data in this rendering algorithm. So there's nothing else, like no part of the interface is dealing with anything else, right? Blender is a really general tool that does a lot of different things, and this is really, really focused. I think that is the main comparison. I actually haven't looked at how Blender does volume rendering, and I don't know if, so typically when you see images of this sort of quality level, I don't know if Blender can do that interactively in viewport or not. I would be curious to see that. I'm sure it's possible, but typically I expect to see that from a sort of offline render that takes longer. I would suspect that our mode is a little bit faster than what Blender does interactively. But again, I haven't done a serious comparison to see what Blender does with volume data.
Dan, could you say a bit about, are you using CUDA? What's the underlying technologies? CUDA, Vulkan, whatnot?
Yeah, so interestingly, it started out as CUDA, and people wanted to be able to run it on their MacBooks. So I ported it from CUDA to OpenGL, and an interesting byproduct of that was that it actually ran faster on the OpenGL side. So currently the underlying graphics library is OpenGL, and that is part of the weird quirk where on Windows, at least, you have to have an actual display device attached so that you can create a window before you initialize OpenGL. So you can't run this in like a headless mode on any Windows platform. You can do it on Linux. And on Mac OS, OpenGL is already deprecated. So the next move of this type of app is to go toward more modern graphics APIs like Vulkan. There's also currently, it's all written in C++, and we have a package, a Python package that is able to load all sorts of volume data. However, the interop between that package and this tool is non-existent. So this tool only currently opens a few file formats. It would be very interesting to be able to just do all of your data loading and even potentially some pre-processing in Python and then hand that Python data directly into something like Agave. I think that actually is a very interesting future direction because it just opens up a lot more ways of getting volume data into this system.
David, did you take your hand down?
Yeah, you kind of stole my question, but it's okay. Thanks.
All right, Greg.
I have like 2.5 questions now. The half question first, because it's just based on what you just mentioned. Yeah, I would love to be able to, in a Jupyter notebook, for example, be processing my 3D data volume, and then in my last cell, say, take this in-memory 3D array, send it to the GPU, and launch an Agave window, just entirely from the Jupyter window.
Yep.
That would be, for me, like the ideal way of getting into getting the data into the client, and instead of having to write it out as like a multi-channel TIFF and then open it myself.
That's right. Yeah, that's exactly right. And I would actually say just one comment on that. Stay tuned. That may show up sooner than later.
Sweet.
We're starting to prototype things along those lines.
Great. And then one of my full questions, and I think Ulrike also mentioned this in the chat. I have some data sets that I would love to look at that are combinations of intensity data, like a proper microscope image, and a 3D point cloud, detections of 3D locations within that volume. If I understand correctly, there's no geometry in here yet. Your annotated data are binary images that have been added to the TIFF as a separate channel, right?
Yeah, that's
okay,
right.
So then what's your recommendation for point clouds? Like I know I could just say, all right, find the closest approximate voxel, let that be a 1. Would a bunch of sparse on voxels distributed throughout the image render well, or should I dilate them, make them squares or spheres? Like, what's the best way to make point cloud data work in this with the current features available in Agave?
So that sounds like it depends on the sparsity of your point cloud data with respect to the volume grid size. So you're either going to have to dilate them, or you're going to have to dilate them to make them fit the volume grid. Yeah, if there are multiple points within one voxel, then you have to find some way of averaging them. I guess you would just do some kind of, convolution or just plain averaging. But yeah, at this point, just to use it in this current iteration of the app, you're going to have to voxelize it in some way. But yeah, I think the direct, simple way would be the first thing I would try.
Cool. And then my last thing was, I guess, more of a comment where Stefan was asking about really big volumes that are maybe outside of a single GPU volume. And you were mentioning, well, you can save out your rendering as a Python script. And if you can, I do a lot of distributed computing on GPU cluster. So I can imagine running a separate instance of Agave on each GPU to render blocks in the image. But I guess if you're trying to render one image of a very big volume, and you're doing it in a blockwise way, the ray tracing is not a purely local algorithm. So you're going to have all kinds of seams issues, and probably also perspective issues too. Like, even if you put everything in the right orientation relative to each other, when you assemble it all into one image, it's probably going to be pretty obvious that there are lighting differences at the seams and stuff like that. So I guess I'm maybe just seconding Stefan's comment that bigger volumes would be great, and a version of Agave that can access multi-GPU memory for a single rendering would be pretty sweet.
Yeah, so there's a recent addition in Agave right now where if you're running it in Linux in a headless mode and you're on a multi-GPU machine, you at least have the choice of selecting which GPU. So if you have one lower powered GPU and one higher powered, you can actually tell it to use the other one. Probably for volumes that are too big to fit in memory, there's a initial brute force approach where I would load only a portion of that volume and then sort of page it in and out. That would be really slow to render. That's going to turn this thing from an interactive tool to a non-interactive tool very quickly. But it would at least allow it to function with that kind of larger data. And then probably in order to distribute it, you would not have each machine have a portion of the volume data. You would probably have each machine work on a subtile of your rectangular pixel grid of your final image output. So,
and page in the portion of the data it needs for just where the ray is at the current time. Okay, so each worker sees the whole data set, but it's only rendering a portion.
Right.
Yeah, okay. Okay, last comment, I promise. Then this one I think is pretty easy, but like, what portion of the GPU memory can the raw image data occupy? Like, can I fill up 90% of the GPU memory just with the image data, or only 50% because it needs the other 50% for rendering?
Yeah, so I haven't done the exact measurement of that. So the amount of memory that's required for the just rendering part of it is generally going to be much, much smaller than the volume data itself. It's just going to store a few buffers that are the size of the pixel dimensions of your window. However, you're also sharing GPU memory with everything else that's happening on your workstation. So in that sense, I can't give you an exact percentage of GPU memory that's consumed.
Cool. Thanks very much for all the info and for a great tool and a great talk.
Sure.
So should we maybe just to finally say something also to the single or to the point data, what Greg already mentioned in his question. So I think there would really be a demand for such a visualization tool also in the browser. So meaning because there are lots of people doing single molecule localization microscopy or even, for example, also paint. So this is really definitely something. If you could put this on your list for future endeavors, which would be, I guess, highly be appreciated by the community. I'm just saying if you also could get into that space as well. So it's just a comment. Thanks.
Okay, well, thank you so much, Dan. This was very informative, and I'll try to get, maybe if we can get the slides so that we'll people can take a look at it, and that will include your contact information, because it sounds like there's a lot of interest here in the general technology, as you know, and maybe we could reach out to you further. In 2 weeks from now, we're going to have Jefferson Amstutz of NVIDIA talk about the Kronos Inari API, which is kind of, you know, Kronos handles OpenGL and Vulkan and a lot of these other open standards. So they've come up with a higher level scientific visualization API that hopefully can be portable and very efficient and do state-of-the-art rendering. And you know, maybe in the future also, like Dan would make use of it for some aspects of Agave. In any case, that's in 2 weeks from now. And at that, we'll close the meetup. Thank you all for attending.